Hello everyone. Today we are going to study Chapter 4 Animal Kingdom. When you look around, you will observe different animals with different structures and forms. As over 1,000,000 species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. 4.1 Basis of classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory, or reproductive systems. These features are used as the basis of animal classification, and some of them are discussed here. 4.1.1 Levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates. That is, they exhibit cellular level of organization. Some division of labor activities occur among the cells. In coelentrates, the arrangement of cells is more complex. Here the cells performing the same function are arranged into tissues. Hence is called tissue level of organization. A higher level of organization, that is organ level of organization, is exhibited by members of Platyhelminthes and other higher phyla, where tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, mollusks, echinoderms, and chordates, organs have associated to form functional systems, each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in Platyhelminthes has only a single opening to the outside of the body that serves as both mouth and anus, and is hence called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory system may be of two types, open type and closed type. Open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it, and closed type, in which the blood is circulated through a series of vessels of varying diameters, arteries, veins, and capillaries. 4.1.2 Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical. That is, any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organism into two identical halves, it is called radial symmetry. Coelentrates, ctenophores, and echinoderms have this kind of body plan. You can see it in figure number 4.1a. Animal like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. You can see it in figure number 4.1b. 4.1.3 Diploblastic and triploblastic organization. Animals in which the cells are arranged in two embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals. Example, coelentrates. An undifferentiated layer, mesoglia, is present in between the ectoderm and the endoderm. Figure number 4.2a. Those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and the endoderm are called triploblastic animals, Platyhelminthes to chordates. You can see it in figure number 4.2b. 4.1.4 Coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity which is lined by mesoderm is called coelom. Animals possessing coelom are called coelomates. Example, annelids, mollusks, arthropods, echinoderms, hemichordates and chordates. Figure number 4.3a. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called pseudocoelomates. Example, Aschelminthes, figure number 4.3b. The animals in which the body cavity is absent are called acoelomates. Example, Platyhelminthes, figure number 4.3c. 4.1.5 Segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomenon is known as metamerism. Notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates, and those animals which do not form this structure are called non-chordates. Example, Porifera to echinoderms. Classification of animals. The broad classification of Animalia based on common fundamental features as mentioned in the preceding section is given in the figure 4.4. You can see it here. I am trying to read this for you. Kingdom, Animalia, multicellular, level of organization, cellular level, symmetry, mostly asymmetrical, body cavity of coelom, acylometa, phylum, Porifera. Then tissue or organ system level, it is divided in two hubs, radial and bilateral. Radial, acylometa. Examples of acylometa is Coelentrata or Nidaria and Tulophora. Bilateral. Bilateral also divided into two hubs, without body cavity, acoelomates, Platyhelminthes, with false coelom, pseudocoelomates, Aschelminthes, with true coelom, coelomates, and coelomates are also divided in parts, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. Okay. Echinodermata exhibits radial or bilateral symmetry depending on the stage. Okay, so in this figure we know that broad classification of kingdom Animalia based on common fundamental features. Okay, the important characteristic features of the different phyla are described. 4.2.1 Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetrical animals. Figure number 4.5. These are primitive multicellular organisms and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute pores, ostia, in the body wall into a central cavity, spongocoel, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, and removal of waste. Guanocytes or collar cells line the spongiocele and the canals. Digestion is intracellular. The body is supported by a skeleton made up of spicules or spongin fibers. Sexes are not separate, hermaphrodite, that is, eggs and sperms are produced by the same individual. Sponges reproduce asexually by fragmentation and sexually by formation of gametes. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. Examples: Cycon, Scypha, Spongilla, freshwater sponge, and Euspongia, butt sponge. That is it for today. We will start from the next phylum the next day. Bye. Take care.